Good morning. Okay, so I'm going to take you on a quick tour of lab and show you some of what I've been working on today. All right, got my ID card and we're going to go in. All right, so here's what happens when you see when you come into lab. These are all beautiful journal covers of different studies that this lab has participated in. They study a lot about fish vision and cephalopods, as well as really cool stuff about stomatopods, which are mantis shrimp. They are amazing. You should look them up. All right, so let's walk on in here. Some of the people in lab coming down here. Posters of scientific work they've done in the past. My office is right there. So I want to tell you what I've been doing for my first couple of weeks in lab. So first thing when I got here, other than just kind of getting oriented, I was given some specimens of a couple of cephalopods so that I could dissect them for imaging. So one is a bobtail squid and the other is a pajama squid. So these were specimens that were collected several years ago for another study, and I'm reusing them, giving them more scientific life, which is great. It means less animals we have to collect from the field, that we can reuse them for a couple of experiments. So what am I doing with them? Well, I'm doing a dissection where I remove as much muscle and tissue as I can. So take the mantle off, take the arms off, take off most of that buccal mass, the big ball of muscle that controls the mouth, the beak, and everything inside. And I even take off the outer portion of the eyeball. So I'm just leaving the retinas, and then the brain sits right between the retinas. So you've got retina, brain, right? And then just a little bit of the upper part of the buccal mass, that big structure that holds the mouth below. So we get it down to as reduced as we can and put it in a very special MRI machine. Okay, so magnetic resonance imaging is a way to image soft structures in great detail. Some of you might have had an MRI done, like to look at a joint, even maybe to look at your brain if you had like a head injury or something that people were concerned about. So magnetic resonance imaging, the kind of MRI that you might get as a person is done in a big magnet, and the magnet strength is measured in a measure called Tesla. Okay, so a human MRI machine could be anywhere from 1.5 to 3, maybe even 5 Tesla would be a particularly strong magnet. The MRI machine they have here at University of Queensland is the most powerful MRI machine in the Southern Hemisphere. It measures at 16 Tesla. That's more than 3 times more powerful than anything you'd put a human being in. And the samples have to be a lot smaller to fit inside this magnet. You couldn't fit a human being in there anyway. The reason it's so strong is to image small samples like mouse brains, or in my case, cephalopod brains. And it gives you very high resolution images of very small objects. So my little 1 centimeter across bobtail squid brain, or my 1.5 centimeter across pajama squid brain, went into the MRI machine, were scanned for about 4 hours to produce high resolution images. All right, why are we doing this? We're doing this because we want to understand, and that's me, Wen-Sung Chung, who's in the lab, as well as some other researchers, want to understand how cephalopod brains are specialized according to the animal's different lifestyles. So you can imagine an animal that is nocturnal might use its visual system less and like its chemosensory system more, right? An animal that's diurnal, opposite, more visual use, right? And so we can look at the visual parts of the brain and be like, oh, the visual part of your brain is very big, expanded. That must mean you are specialized for vision, right? So we can look at different parts of the brain and see what they might be expanded or specialized for by looking at the anatomy. The anatomy is very poorly understood in a lot of these animals. And so it's exciting to get to be part of that. So the images we did last week of bobtail squid and this week of pajama squid are the first times those brains have ever been imaged like this. So anything we learn is going to be exciting and new. All right, so let me show you some of those images. All right, so this is the bobtail squid brain. What you're doing is you're looking at my MRI images cut in 3 different sections through the brain. So we can see it sort of like, you know, on all 3 planes at the same time. And I can move through. So you see this top section, I'm moving through the section and seeing all these different structures as we move through. So let me show you a few highlights. So here we're looking at one section of the MR image that goes right through the middle of the squid's head. So we can see the retinas on either side, and then in between the two eyes is that big gray area is the brain. And then right in the middle of the brain, that green spot I've highlighted, that is actually the esophagus. All right, so now on the right side, you can see one optic lobe is highlighted in yellow. The optic lobes are symmetrical. There's one on each side. I only highlighted one. The optic lobes are really important for visual processing. So finally, in pink at the top, that is the vertical lobe. The vertical lobe is thought to be important for higher order cognitive functions like learning and memory. There are other parts of the brain too. I just wanted to highlight a few for you. I am learning to identify probably a couple dozen of different brain structures in the squid brain. This is the wet lab space where people can do things like examine specimens. Like I said, they do a lot of vision work in here, looking at vision of fish and stomatopods and cephalopods is particularly of interest. Personally, I have worked right here at this microscope. This is where I've been working to do my dissections. So here's a dissection in progress. You can see the little bitty creature in here. It has two of the arms still attached that I used to help pin it down so I can work on it. The eyes are still there as well. And under a layer of cartilage right up here at the top lie the optic lobes on either side tucked behind the eye. Then here in the middle, that is where the vertical lobe sits. Okay guys, I wanted to answer a few of your questions. So one question that I was asked after my first couple of videos was if any of the cephalopods have venom that could kill you. The answer is yes. In fact, one of the species that I may be working with, the blue-ringed octopus, it's a really common animal in this part of Queensland. It produces a venom called tetrodotoxin. Actually, bacteria that live in a symbiotic relationship with it produce the tetrodotoxin. That tetrodotoxin is released into the saliva, and it is a paralytic. So if you are bitten by that octopus, it's quite small and very pretty, and so sometimes tourists pick it up, which is a bad move. And if you're bitten by it, really the way people might die of tetrodotoxin is that they would stop breathing. So I have gloves, and I have a cephalopod expert who's going to be with me so that I don't get bitten by a blue-ringed octopus. Okay, but all octopuses are venomous, and probably all cephalopods are capable of producing some kind of venom. They act in different ways. There's different kinds of proteins and peptides and chemicals that create that venom. But yes, that helps to paralyze their prey so that they're able to eat it. Because again, remember, their esophagus runs up through the middle of their brain, so you do not want something that could fight back while you're swallowing it, right? Whatever it is, it's got to be dead or paralyzed or motionless. And then also that big beak acts like a raptor beak. It can tear off chunks of flesh. That also will help it swallow its food without injuring its brain. All right. Another good question I was asked is, do octopuses or other cephalopods feel pain? All animals, since damage to their tissues. Now, the thing is that what we call pain is more than just that sensation of damage to your tissue. It has an emotional component, and it can be experienced differently depending on the circumstances you're in. And we all know this. Like, you've had cases where, you know, you hurt yourself and you don't even notice because you're focused on something else, you're doing something else, right? You bump your knee and you don't even realize until you see a bruise later and you're like, where did I do that, right? Or times when you've got really high adrenaline going, right? And so you don't even feel the pain until later on. Other times you might be tired, cranky, run down, and the smallest thing feels really bad, right? Because of other stress you're under. So that emotional side of pain we don't know what other animals feel. That's why we always anesthetize, right? And do our best to have some kind of pain relief so that the animals do not experience undue stress or trauma. And that's why actually all of the work we do goes through review by an ethics board. We're doing everything we can to make sure that animals don't experience stress or pain in our care. Okay, another great question that someone asked me is, why are you interested in this particular type of science? I'm interested in this because I am a curious person. I want to understand how all these different biological functions work. It's one of the joys of my job that I get to just learn and play and find out new things and solve little mysteries in the biological world. So I'm really curious about these animals. We know so little about them. And I want to better understand how they evolved, how they live their life. And we'll talk more about that. I really want to tell you guys some interesting evolutionary facts in a future video. So I hope we'll keep coming back to that question as we go. But there's another question you didn't ask that I want to answer anyway, which is, how do I know how to do this stuff? So I just talked to you about my doing these dissections of these bobtail and pajama squid, and then doing MRI of them, and now analyzing the MRI. And there's sort of two sides to that answer. One is I do have a lot of past experience doing dissections and surgeries on other animals due to my job, right? I've examined brains in lots of different animals, and so therefore I have done dissections, I've done surgeries, and I know I have some good surgical skills of doing, manual dexterity and doing small things and dissecting under a microscope just due to practice, right? That stuff is hard at first and you get better at it over time. So some of that is just me using those skills that I have from before. I've also done image analysis. I've even done image analysis of MRI before on human MRI. But the other side is sometimes I don't know what I'm doing. So my first dissection on the bobtail squid, I accidentally took a big chunk out of the brain. There's a big hunk missing from the optic lobe on one side. Because I was doing the dissection, I was removing some cartilage. I just got a little over enthusiastic and took a chunk of brain out. And then all of a sudden I went, and it was too late, it was gone. So I still make mistakes. Same thing with the MRI. I was working on tracing outline instructions on the MRI, and when Sung came into my office and looked over my shoulder and was like, well, why are you doing it that way? I was like, what do you mean? And he pointed out to me I had misdrawn a structure, and I was like, oh, okay, great, thanks. I made some wrong assumptions about the anatomy. He got me back on track and I started tracing again. And I just had to redo a couple hours work. That's part of the process. So I am doing my best to apply old skills, and I am also learning all kinds of new skills as I go. So I guess my lesson there is practice, practice, practice. Be patient with yourself, be patient with others, and you'll get it. Right? Don't be afraid of making mistakes. The only way I'm learning how to do all this stuff is because I've made a lot of mistakes, and I just try and learn from them.